Okay guys, buckle up because this is going to be a lengthy set of notes. Hopefully not a whole period, but we'll see where we can get. We have 40 slides, so I believe we can get through it definitely in the time we need, okay? So you were supposed to do this spell work. I believe it's in your notes. There are 3 tenets, 3 statements that are facts for cell theory to exist. And you need to remember what those 3 are. You need to write them down in your notes. You can see them there at the top. Hopefully you've looked them up already. And then you're supposed to put a star in your notes next to the one that you think we're going to be studying in this unit. And if you don't remember what the title of the unit is, well, look at the top of your page, page 1. Mitosis and asexual reproduction notes. Make sure you get that right. Make sure you don't guess. Look it up later if you don't want to make a mistake, okay? So why do cells need to divide at all? Why can't they just, a baby just be born and that's it? Well, it's pretty obvious. You can't stay the size of a baby. You have to grow. The cells themselves can't grow bigger and bigger and bigger. That would look very strange. You'd look like a big baby. Some of you act like big babies, but imagine a baby the size of a teenager or an adult. That's some freaky stuff right there. So the cells don't grow that much. They have to grow a little bit and then they have to replicate themselves, right? So, and that's because when a cell grows in size, its cell membrane starts to lose the ability to hold everything in. It's like a balloon, stretching and stretching and stretching, and finally, it's gonna blow, okay? It's gonna pop. So cells typically don't do that. Instead, they replicate themselves. So it's the volume inside that needs to be compensated for. Something has to happen to fix that growing volume, okay? So it's growing at a faster rate than the surface area can hold. A balloon growing at a faster rate than the volume inside can contain, okay? So the cell membrane becomes too small to hold in all that stuff. So once a cell reaches a certain size, it won't transport things well across the membrane, for example. And it'll die, it'll burst, it'll pop, you know? Instead of doing its function, it will die. And it has to do with the ratio of cell membrane to volume of cytoplasm inside, okay? So it has to divide, it has to replicate itself, or the cell will die. So why else do cells divide? Growth of multicellular organisms like you and me, okay? And plants, and worms, and sharks, and dolphins, and elephants, and mice, and everything else in between, okay? Multicellular organisms. The only way for them to increase their size is to increase the number of cells. Like I said, otherwise you'd just be a big baby. Replacement and repairing of damaged tissues. Ouch, I got a boo-boo. How does your body fix itself? It makes new cells. Mitosis, okay? Mitosis is the word that we're looking for here. Even when multicellular organisms stop growing, their cells have to be replaced, excuse me, as they die or become damaged. Red blood cells only live for 3 days in your body and they have to be replaced every 3 days. So plants have to grow to produce their parts that we learned about, their flowers and their stems and their leaves and their fruit and their roots, okay? And then of course asexual reproduction, which we will finish up these notes on at the end. We will talk more about asexual reproduction, okay? But to start off, just as a definition as to why cells need to reproduce themselves, okay? Certain organisms, not mammals, humans or mammals, so not humans, can reproduce by forming an exact copy of themselves in several different ways. And this form of reproduction is called asexual because it involves only one parent. Okay? So here's an example, and here's a question in your notes about that. So make sure that you get this question answered either now if you have a chance or later, okay? After learning about flowers, we learned about the anatomy of plants and flowers, right? Which we're going to have a quiz on in the next few days, if not maybe next week sometime. So just know that you still need those notes, the leaf anatomy sheet, because we're going to be having a quiz on that, very short quiz, okay? So after learning about flowers, how are you sure that this picture is showing asexual reproduction? How do you know that this is not sexual reproduction? Okay? So think about that, write the answer down. Okay, maybe even for later at homework, when you're going over these notes like you're supposed to. Okay, your homework tonight is to go home and read these notes for 10 minutes and fill in any blanks. You'll notice at the top of the notes, no grade if name is missing. So I've been getting a lot of no names. And no grade if any questions are skipped over. You don't get a grade. I'll send it back to you until everything is done, okay? That's what I'm looking for. So how is the nucleus involved in cell division? So in order to understand what's going on, we have to understand what's going on inside the nucleus itself, the brain of the cell, right? Remember? With the DNA inside? So the nucleus controls the activities, making new organelles, transporting things in and out, making proteins to be transported in or out or to make new things, okay? But it also has these things called chromosomes in them, which replicate so that the cell can split in two. You have to make doubles of everything so that you can have replication, okay? Like a photocopy, almost. And they're composed of DNA, deoxyribonucleic acid. Now, I told you in the past that in the future, which is now, poof, you're in the future, you had to know what DNA meant and how to spell it. So there it is. Write it in your notes, put an asterisk, that's how you say that word, asterisk, next to it, okay, a little star, so that you know that that's important, and you have to know how to spell that, okay? So inside the cell is the nucleus, inside the nucleus is the chromosomes, and those chromosomes are made up of tightly wound DNA molecules, deoxyribonucleic acid molecules. And the DNA contains the hereditary information so that both parents can code for their particular species of, type of organism. So 2 dolphins will make a dolphin. 2 cats will make a cat. House cats. Not a cat and a lion. That won't happen. They're different species. Okay? DNA consists of long chains of nucleotides. Remember what those are? Remember macromolecules and polymers and monomers, a bunch of monomers put together make a polymer, a chain. Well, that's what this chain is. This double helix of a chain, these are your hydrogen bonds holding together the subunits. The nucleotides, organized into a twisted ladder. So if you took a ladder like that and you twisted it, that's the organization of a DNA molecule. Chromosomes. Chromosomes are the tightly wound up DNA strands like this. They look like that. Okay. The 2 identical strands, one from the actually, I take that back. When the cell is getting ready to split into 2, it replicates itself. It replicates a copy of the DNA. So this is one copy and this is the other copy, and they're held together by this thing called a centromere. So you're labeling this in your notes, right? So each of the copies are called sister chromatids. This whole thing is a chromosome, and then the sister chromatids, and they're again held together by a centromere in the middle. You've got longer arms, shorter arms, sister chromatids. The whole thing's called a chromosome, and the ends of the chromosomes are called telomeres. Centro, tele means end, telomere. So make sure you label that up. If you miss any of these, you have to come back on your own, okay? Perfect time to do it is tonight for your homework. Yes. How is DNA packaged into chromosomes so the cell can replicate itself? Let's watch a short video. It's pretty cool, actually.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing.
We'll learn these words later.
As we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, so what they showed was how the DNA here gets coiled up into these chromosomes when a cell is getting to replicate itself. Normally it's not like this. Normally it looks like a bowl of spaghetti, okay? Like a bunch of strands of yarn all in a bowl, okay? But then when it gets to replicate, they have to organize themselves. Imagine taking a bowl of spaghetti and making braids out of individual, maybe 3 or 4, making braids and then organizing those braids into pieces like here. That's exactly what the cell does by itself with the help of proteins that organize it. It's really quite amazing. Miraculous, really. So you have to label this in your notes, okay? So the cell membrane, we know what that is, right? That's the wall, not the wall, sorry, those are plants. Plants have cell walls. It's the barrier between the inside of the cell and the outside of the cell. I mean, this happens in plants too, but right now we're focusing on an animal cell. So the nuclear membrane, it's very similar to the cell membrane, but it's a lipid bilayer that contains the nuclear material inside the cell. Chromosomes are inside the nucleus, and here's a blown up version of it with the telomere, the centromere, like you just labeled. The chromatin, which is all wound up in here. The condensed histones, so we saw that happening in the very beginning of the video. The beginning of the video started with this and then condensed it into these bundles, and then condense those bundles even into more bundles, and then condense, which is chromatin, and then condense the chromatin into the chromosome. Whoa, crazy. So those are the proteins that are making the histones, okay? And again, that's the double helix, the DNA double helix, the twisted ladder of nucleotides, the base pairs. The sugar phosphate, let's move that out of the way. Okay, sugar phosphate backbone and the nitrogenous bases, just like what we learned in the biochemistry unit about the nucleotides, pairs of them coming together, and we'll learn more about what all that means, all those letters in there, in the genetics unit, which is coming up later, not now. So you should have everything filled out on that. Again, if for some reason that you are a little slower and you miss it, then you're gonna have to come back to that and fill it out later, okay? Or look at somebody else's paper later. Okay, we're on slide 12, the nucleus and the chromosomes continued. So for non-dividing cells to conduct their day-to-day activities, the chromosomes have to unwind the DNA, okay? They have to unwind it. In this state, the individual chromosomes cannot be seen under a microscope, and that's what I was talking about before, like a big bowl of spaghetti. That's called chromatin. So these are the different, so we start with the DNA. The DNA gets wrapped up into these nucleosomes, okay? And then the nucleosomes condense into this chromatin fiber. The chromatin fiber then condenses. Imagine taking your spaghetti and then wrapping your spaghetti up in a pattern and then folding that pattern on top of each other, and then now you have this rope, and then now taking that rope and bunching that rope up into this, and then that's how you get the chromosome, which is now ready to replicate in a cell, go through a process called mitosis. And so these are what they look like under a microscope, okay? So the chromatin begins to coil up, each strand becomes thicker and shorter until it can be seen under the microscope, and these things that can finally be seen in the microscope are called chromosomes. This is the nucleus before, this is like the bowl of spaghetti. So this is a cell that's not going through replication. These cells are getting ready, almost getting ready to split in half. You see how the DNA now is in chromosomes, and the chromosomes split, and now you're gonna have two cells here. Same here. These are gonna pull apart this way, and you're going to have two cells here. So these cells are in replication mode, these cells are not. Now, all organisms have different numbers of chromosomes. So you're going to fill in these numbers for these different organisms. The roundworm, the rice, yes, rice is a living organism, right? It's a seed of the rice plant. And so they have DNA, and they have chromosomes, 24 of them. We have 46, chimpanzees have 48, and dogs have 78. Canis familiaris, the house dog, I guess so, house cat, house dog, they have almost twice as many as we do. Number of chromosomes does not mean you're more intelligent, all right? It just means that you have different traits to be expressed, okay? I mean, there's a mustard plant that has like 100 and some odd chromosomes, a plant, so 4, 24, 46, 48, and 78. Now, all organisms have a double set. They get one from mommy and one from daddy, to put it very simply, okay? One from each parent. So the total number of chromosomes found in the nuclei of any organism is a double set, double number, and we call that double set diploid, the diploid number. And it's represented by the symbol 2n, okay? So in your notes, diploid equals double. Make sure you write that down. That is on the bottom of page 4, okay? Slide 15, bottom of page 4, that's where we are right now. So you got egg from mom, sperm from dad, and you can see how they've got copies. Okay, and now the zygote is going to be double. Double the egg, double the sperm to make a diploid number. Di means two, right? The diploid chromosome for humans is, you just wrote it down in that little diagram, is 46. And all body cells, skin, bones, brain, muscle, your intestines, your eyeball cells, all your body cells except for, well, the only ones that aren't body cells are sex cells, okay? That's the ones down here, sperm and egg. Those are called gametes or sex cells. These are opposites. These are going to have a diploid number, and these are going to have another number called haploid. Haploid half, H-A-H-A, half haploid. We'll get to that in a second, okay? So you can see a somatic cell, a body cell, you've got double the number, but in a gamete cell or a sex cell, sperm or egg, you're going to have half the number. Because otherwise the baby would have too many chromosomes and they wouldn't develop properly. If you mess with just one chromosome number, just one, you get something that could be Down syndrome. If you know anybody who has Down syndrome, that person has, I forget now, is it an extra or a missing chromosome? Yell it out if you know, I forget. But we'll learn more about that in the genetics unit. And so this is what human beings' chromosomes might look like. I'm not sure if you can tell, but there are pink and green, or purple and green. The green ones, chromosomes from the father. The pink ones are chromosomes from the mother. And you have a total of 46, including the X and the Y chromosomes. And this would be a male because it has X and Y. A female would be X and X. And that's what makes the female different from a male. It's just those two chromosomes right there, whether it's XX or XY. And that's determined by the sperm, believe it or not. The sperm determines whether the baby is going to be male or female because they have X and Y. So to maintain that constant number from one generation to the next, when the sperm and egg, the gametes, when they come together, they must carry a haploid or half, one set, a single set of chromosomes. This half number of chromosomes, like I said before, is called haploid. And it's represented by the symbol N, opposite of 2N, okay? Just like there's a little 1 there, right? An invisible 1, just like, and you notice it says, just like in math, X equals equal to 1X. The 1 is invisible. It's a given. You don't have to write it down. So all the sperm and egg will have half the number of chromosomes so that when they come together, the offspring will have a diploid number of chromosomes and be able to function as a normal member of that species. So it works together. It works perfectly. It's a perfect system, okay? When it works perfectly. So chromosome numbers. So the haploid chromosome would be 23. So half of 46 is 23. So all of our sperm and egg will have the haploid number, and then when they come together, the baby cell will have the diploid number, okay? So here you can see one copy of each chromosome in the sperm, and it's going to be one of these. So 50%, it's going to be one of the sex chromosomes, either X or Y, and then whichever one it is, whichever one meets up with the egg and fertilizes the egg, then the baby will be male or female. Because the egg will always carry just X. If this is an X sperm, you have a female baby. If this is a Y sperm, you will have a male baby. It's pretty cool. So using the information that you were just given in the notes, complete the following statement and table. I'm not going to give you these answers. It's really simple. They give you the hints here, okay? And then you would just determine the other numbers based upon the information that we just learned about and filling in that table and that sentence, yeah. Okay, we're on slide 21, so we are on page 6. How does cell division take place? 2 processes, and then you have 2 new cells from a parent cell, okay? Mitosis and something called cytokinesis. Mitosis is the actual process of doubling the DNA and the other organelles. And then cytokinesis is the breaking apart of the 2 cells, and now you've got 2 cells from one. The one cell grows, gets too big, goes through mitosis, and then goes through cytokinesis, and now you have 2 cells from the one cell. That's how it works, okay? You're dividing the nucleus. Cytokinesis is dividing the cytoplasm, right? And then when neither one of these is happening, when the cell is done dividing and just resting and doing its job in the body, that's called interphase, okay? And that's the time between one cell division and another cell division. So one cell will become 2, and those 2 will become 4, 8, 16, 32. Baby's born. Well, it had to be a lot more than 32, but you know what I mean. And that's called the resting phase. It's the longest phase. Interphase is when you have the bowl of spaghetti, right? And that's the DNA just existing in the nucleus. And all the other organelles would be out here as well. And there's no division taking place. So you have to fill out all the stuff you see on that on this cell cycle diagram in your notes, okay? And you've got nice blank spaces there, including this part. I didn't put blank spaces here because they'd be too fat, so you have to fill in these blank spaces. Ready? Okay. So this is the cell cycle diagram. So this whole part here is that resting phase we just talked about, called interphase, where there's no cell mitosis happening. Okay, it's the resting phase. Most of the cell, as you can see, you have to think of this diagram like a clock, okay? Like a clock. And this is the main part that there's no cell division occurring, okay? You have G1, S, and G2 phases. The G1 is where the cell's growing, so G for growth, I guess you could say. S, DNA replication. So S is for synthesis. The DNA in the nucleus is replicating itself. And I'll show you a video of what that looks like in a moment. It's pretty wild. And then preparation for mitosis, G2 stage. So now the cell is growing. It said, whoa, we're getting kind of big. Better replicate the DNA. Better get ready to split. And that's what's happening. And then the splitting happens right here, okay? So you're going to have to memorize the stages of mitosis, okay, in order. And it's really kind of easy to remember because there's a way to say it. So it's prophase, metaphase, anaphase, and telophase. PMAT. It's like the thing you put down for a baby puppy when they haven't been potty trained yet. A PMAT. Yeah, that's how you remember it. I know it's goofy, but if you can come up with something better, go for it. PMAT, okay? Prophase, metaphase, anaphase, telophase. And then the cell splits. And now you've got 2 new cells. And then it goes through the whole process again. This whole thing, from prophase to telophase, is called mitosis, and then cytokinesis. So and this whole region here is the cell division part, okay? So spell these correctly on that diagram. Prophase, metaphase, anaphase, telophase. And that's when the cells are splitting apart, the nucleus is splitting apart, and then you get the cell splitting apart. If you, for some reason, you are needing to work at a slower pace, by all means, again, I'll say it, this is the third time, go back and look at this later. It is your job as a student tonight for homework. All right, you're going to do these for homework as well. So these are your test yourselves, slides 23 and 24. And then you have to, so mitosis, PMAT, right? These are the stages, and this is what happens during each stage. This is an extremely important diagram that you have to fill out in your notes. So I'm going to put the answers up for you for the boxes. So in interphase, you have the bowl of spaghetti, the chromatin, right, in the nucleus, in the nuclear membrane. And the centrioles, the things that pull apart the chromosomes, are just out with the other organelles, okay? Then you enter mitosis. Once you leave the interphase, you enter mitosis. PMAT, P-M. We're going this way, sorry. A, T, PMAT. Prophase, metaphase, anaphase, telophase. In prophase, you have the chromosomes now, so the bowl of spaghetti has turned into the chromosomes, and the nuclear membrane breaks down. And these spindle fibers that come from the centrioles line up, start lining up the chromosomes along a center line, okay? So that's prophase. In metaphase, meta meaning middle, meta is just MM, meta middle, okay? All of the chromosomes are lined up right down the middle of the cell. And these spindle fibers are going to attach themselves to these centromeres, and they're going to pull apart the chromosomes in halves. Here, okay? They're going to pull them, so they grab onto the centromere, and they pull them apart to opposite poles of the cell. And so metaphase, middle. Anaphase is when they start pulling apart. And then you get this thing called a cleavage furrow right here. This is called cleavage furrow. And that's where cytokinesis is going to occur. That's where the cells are going to split into two. And then the nuclear membrane forms around the chromosomes again, and they go back into interphase again to the resting state. And you have two cells. They're called daughter cells from the first cell. Okay, so we're going to watch this video. So if you missed any of this, you have to come back to it. Okay, you have to study this for the next test. All right.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis.
Okay. How does cytokinesis, speaking of that, that's why we ended, differ in animal and plant cells? Well, the only real difference is that in a plant cell, you get this cell plate that forms instead, okay? And we already talked about cleavage. Cleavage is the cleavage furrow, and it's a contractile ring that breaks the 2 cells apart. But in plant cells, they have this cell plate that forms the new cell wall, and that's what helps separate 2 plants to plant cells during cytokinesis. That's why it's different. The only difference between animals and plants is that cell plate, okay? You may need to know that on a multiple choice question. What is the difference between animal and cell and cytokinesis? And you'll have to say choose cell plate. So what's the result of mitosis? Okay, every time a cell undergoes mitosis and cytokinesis, two identical clones, daughter cells, are the result, okay? With the same number and same kind of chromosomes. This way, multicellular organisms can grow by increasing their number of cells in their bodies and repair damaged cells, okay? And some organisms can even replicate to form identical offspring. Not, again, humans, not mammals, not humans, okay? And a way to remember, because you're going to have to learn another word that sounds like mitosis in the future, is mitosis, okay? So if you hurt your toe, how does your toe fix itself? How does it repair? How do you grow? Mitosis, okay? So remember mitosis. Question in your notes. If a human body cell divides by mitosis, how many chromosomes will the daughter cells have? And it's identical to the parent cell, okay? Parent has 2, daughter cells have 2. I don't mean daughter like child. I mean when a cell replicates and splits into 2, those 2 cells are called daughter cells. Okay. So how does the rate of cell division maintain homeostasis? Different types of cells divide at different rates. So red blood cells, for example, must divide quickly, but there are other cells that divide at different rates. So in order to maintain an organism's ability of to remain stable, that's what homeostasis means. Remember, you were supposed to define it in that last set of notes, right? Different cells have different functions. So nerve cells, skin cells, liver cells, they all have different time periods. So nerve cells might not replicate in 70 years, okay? Skins, because you have them all already. Skin, and they live a long time. Skin cells every 20 hours. That's why you're constantly sloughing off dry skin in your body and from your hair. If you did this in like a beam of light, like sunlight through a window, you'd see a bunch of skin fall off. That's normal. Little flakes, almost like dandruff from your arm or whatever. That's normal. Liver cells, about every 2 months, 150 days. And it can repair itself. So you can, that's why you can get a transplant. You can even donate part of your liver or, like you do a kidney to somebody who can be a match. And you would heal from that because of mitosis, okay? All these different rates are used to maintain that stable body condition we call homeostasis. Now, what happens when the cell cycle malfunctions? Well, the cell cycle determines the rate of cell division. The cell cycle is closely monitored and regulated by the body. Each cell contains different genes to control the length of each stage. Sometimes the body loses control of that and cancer can develop, okay? Now, cyclins are groups of proteins that regulate the timing of the cell cycle in eukaryotic cells. And this diagram down here is just showing how a cyclin comes in, and then when it's used, it just gets destroyed and more cyclin gets produced. So, and they're used for determining when those cells should divide or not. So cancer results from abnormal, rapid, uncontrolled cell division. And that's a result a tumor can form. This is what tumors are. Clump of cells that develops. So you have this cancer, a normal cell turns into a cancer cell, which replicates wildly. And then you get this malignant tumor. Malignant, spell it correctly, means bad, right? Mal in Espanol means bad, right? So, and benign means not bad, kind. Benign really means kind. So sometimes they can still be bothersome, but they're not cancerous, all right? They don't invade surrounding tissues. Malignant tumors will spread to other parts of the body and other tissues, and they'll interfere with organ function. That's why this is considered cancer, malignant, and this is not, okay? All right, so you're gonna fill in these at a later time, the answers here, so don't worry about getting that now. You can finish that for homework, okay? So you're gonna have to come back to these notes for homework. What are the different forms of asexual reproduction? Binary fission. That's when like a bacteria splits and grows. Single-celled organisms only. Protists like algae and plankton that are swimming around in the ocean. And plants, you can break off a piece or cut a piece and stick it in the dirt and it will just grow. You can't break off an arm, stick it in the dirt, and the new person grows, right? That'd be kind of creepy. And other organisms can regenerate. So animals that are the hydra, the sea star, a planaria worm, for example, they can have a part of them grow back an entire new body, actually. But vertebrates, those organisms with a backbone, typically cannot do this. Very few can grow back parts. This is called budding. So this is the parent organism, and this is a bud coming off. This will be kind of like an identical offspring that when this breaks off, it'll grow just like the parent will, okay? Sponges can do that. You can take a sponge and chop it up into 100 pieces and throw it in the water, and each piece will grow into a new sponge again. That's wild. A living sponge, you know, not the sponges at your sink. Those aren't alive. Those are synthetic sponges. Binary fission, again, is when single-celled organisms split themselves through mitosis and cytokinesis to become two daughter cells. Mitosis, cytokinesis, two daughter cells. Bacteria, the amoeba, and the paramecium are examples. And this is budding. Like I said before, the bud forms off, okay? And it may separate from the parent or remain attached. Doesn't matter. Yeast does this as well. Yeast is the living organism used for, we talked about this in fermentation earlier, a type of cellular respiration, remember, without oxygen. Sporulation, these are spores from a fungus, these are spores from a fern, and they are special single cells that are asexually produced, asexual reproduction. It's a type of asexual reproduction. And so when the spores enter the air, they land someplace else and just sprout and grow into an identical parent. And then regeneration. So this is a crab claw that was pulled off and is regenerating, right? It's the ability of animals to grow back missing parts. So when they harvest certain crabs, they don't kill the whole crab. They will just break the claw off and the crab will grow back its claw, okay? And this power decreases when animals become more complex. So I couldn't cut the trunk off of an elephant and then the elephant's trunk grow back. It wouldn't do that. And then finally, the last slide. I'm not going to show you that video because we're out of time, but sea stars, As long as you have a central part, will regenerate and grow back into an adult. And newts and certain lizards, right, can have their tails cut off, and the whole tail will regenerate after a certain number of weeks. But if you cut a leg off, the leg won't grow back. It's a survival technique. And that is our note.